So I thought I'd start off with some basics. And when I Googled images of lecturers, most of the images that came up were men. This was the only woman who came up, but I quite like her. She's having similar microphone problems to me, and she's gone for a handheld mic rather than holding the microphone box. So if I just start off by giving you an outline of the things I'm going to tell you about today. So I'm going to give you an introduction to viruses and then tell you a bit about cancer viruses. And then focus on the particular cancer virus that I'm interested in, which is a virus called Epstein-Barr virus. I'm then going to try to explain some of our most recent research and discoveries. And then in the last part of the talk, I really want to acknowledge the funders and the people and family who have supported me throughout my career journey. So if I start off with the question, what is a virus? Well, a virus is a microscopic infectious agent. And the key thing about viruses is they cannot reproduce by themselves. They have to infect a host cell, a living cell, in order to be able to reproduce. This is an illustration of what a virus looks like at a high magnification. The viruses are able to attach to host cells through spikes on their surface. And when they get into host cells, they take over the machinery of the host cell in order to produce more virus particles. So if you just look on the right for now, this is taking you through the main steps in the life cycle of a virus. So the first thing that I should tell you is this is not to scale. Viruses are much, much, much smaller than host cells, but for illustration purposes, I've made it larger so you can see some of the steps that I'm going to talk you through. So the first step, as I mentioned, is the attachment of the virus to the cell that it's going to reproduce in. The virus then enters the cell and releases its genetic material, or the virus fuses with the cell and opens out and releases its genetic material. It then replicates its genetic material because it needs this to be able to produce new virus particles, and it produces viral proteins, some of which are required to form the outside, the packaging parts of these virus particles. And new viruses are then produced and released. Quite often, with a number of viruses, during this process of producing new viruses, the host cell is often destroyed. But some viruses have the property of being able to become dormant and hide in infected cells. And when they're in this form of dormancy, they don't kill the host cell because they need the host cell to stay alive in order for them to stay alive. And cancer viruses often have this property, so they have the ability to hide in host cells in a kind of dormant phase and not actively go into this process of producing new viruses very often. So if I move away from the general introduction to viruses and then think more about what we know about the link between viruses and cancer, we know that viruses play a major role in the development of lots of different types of human cancers. And it's estimated that about 20% of cancers worldwide are caused by viruses. So that means that roughly 2 in 10 cancer cases are the result of viruses. We know that some virus-associated cancers occur as a result of problems with the immune system. So as a result of our inability to control the outgrowth of the virus or the virus infection. But other virus-associated cancers have other contributing factors. So there may be genetic alterations in the cells in which the viruses are hiding, or there might be other environmental factors that push those cells towards developing into cancer cells. So don't panic, there's a lot of information on this slide. I don't expect you to take all of it in. I've just put it up here for illustration purposes to tell you that there are 6 main viruses that are associated with human cancers. And this is really to illustrate that they don't all come from the same virus family. So there isn't one type of virus that drives cancer development. They come from a range of different virus families, and they even have different forms of genetic material. So some of them have DNA, the same genetic material that we have in our chromosomes inside our cells. Some of them have a different form of genetic material called RNA. They also infect different cell types. So for example, there are 2 hepatitis viruses that we know drive liver cancer development, and they infect liver cells. Other viruses infect the cell types in which they end up driving cancer development. That's quite obvious. I'm going to come back to talking much more about Epstein-Barr virus and the cancers that it causes. But that's just to really illustrate there are different types of viruses from different virus families that infect different cells. So exactly how do they cause cancer? So I've explained to you a little bit about the virus life cycle and the fact that they might be able to enter a dormant phase in which they hide in infected cells. So if I just go through what they do to those cells when they're in them, that helps to explain a little bit of why they drive cancer development. So this is the first step. The virus attaches and infects the cell and injects its genetic material into the host cell. Well, this genetic material drives the production of viral proteins within that infected cell. And these proteins have an effect on the host cell. So in the case of cancer viruses, what these viral proteins can do is to force the production of factors within this cell that push the cells to grow in a way that they wouldn't normally grow. In another way of switching on cell growth, they can also push these cells to keep replicating their DNA. So this is the nucleus of the cell that contains all of the cell genetic material. This needs to be reproduced in order to be divided into daughter cells when new cells are produced. And viruses can push cells to keep replicating their DNA and producing more daughter cells in a way which is at a higher rate or in an uncontrolled way to how this cell would behave normally if it wasn't infected with a virus. But what they can also do is something that people find quite surprising. When I've explained my work to bloggers or journalists before, this is the one part that really kind of sparked their interest. And this is the fact that within our cells, we also have mechanisms which can stop our cells from growing. Maybe that's not so surprising. But we also have mechanisms that allow our cells to commit suicide if they sense that things go wrong. And this is a really important protective mechanism in our cells. So they can sense that this is happening and they would trigger their own suicide. They want to destroy the cell because they recognize that there's something wrong with it and they don't want that cell to survive in the body. And viruses often interfere with both these processes, so they push growth, but they also interfere with the mechanisms that stop growth or cause cell suicide. And the net result of this is cells that don't die when they should, when things have gone wrong, and cells that keep growing and keep dividing. And in the case of many cancer viruses, these cells then become what we describe as immortal. And that essentially means what you might expect that it means. They just grow forever, indefinitely. They have the capability of continuous, indefinite growth, unless something should stop them. And that's the hallmark or the first step in cancer development, because that's a really important property of a cancer cell. Cancer cells just keep dividing. Okay, so that's a bit of a general introduction to viruses, hopefully a little bit of information about cancer-associated viruses and how they drive cancer development. So I'm going to tell you now a little bit more about the virus that I'm particularly interested in, and that's a cancer-associated virus called Epstein-Barr virus. And this was actually the first human cancer virus to be discovered. And this virus was discovered as a result of the work of a British surgeon called Burkitt, who was travelling through Africa, and he noted a particular cancer that occurred in children in the jaw, eyes and abdomen in a particular part of Africa, in sub-Saharan Africa. And he noted that this cancer, which is described as a lymphoma, it's a cancer of white blood cells, and we describe those cancers as lymphomas. This cancer actually had the same incidence as where mosquitoes were prevalent. And mosquitoes carry a parasite that causes malaria, so it was also in the same region that malaria was present at high levels. So Burkitt, working together with a team in London headed by Tony Epstein, postulated that there may be an infectious agent that is causing Burkitt's lymphoma. So they went looking to try to find if they could find an infectious agent. So Burkitt sent some of these lymphoma cells to London, and Tony Epstein, working with his technician Yvonne Barr and a microscopist, Bert Akon, who had a very powerful electron microscope that was able to zoom in at high levels to look for infectious agents in these cells. discovered that there was indeed an infectious agent in these cells, and that it was a virus. Later on, a team in Philadelphia in the United States showed that they could actually take the virus that they isolated from cultures of these cells and mix it together with completely normal white blood cells that they'd isolated from donated blood, and the virus could infect those cells and render them completely immortal, so make them continue to grow indefinitely. So this was very good evidence that in actual fact this virus was causing this lymphoma. However, I guess the interesting part of the story, if it works, is that the virus actually isn't carried by mosquitoes. So the original reason for going looking for an infectious agent was the mosquito link. But it turned out actually that it's malaria that seems to help drive the cancer to develop. The mosquitoes aren't carrying the virus, but people that are infected with malaria seem to have a higher risk of developing this particular lymphoma. And I'm going to come back to that a little bit later. So, if EBV isn't an infectious agent that's transmitted by mosquitoes, how is it transmitted? Well, actually, it's passed from individual to individual through saliva. So that's either through kissing or from the sharing of eating utensils or cooking utensils. And the virus has specificity for infecting two particular types of cells in our body. And the first cells that it encounters are the cells that line the upper part of the throat behind the nasal cavity, which is called the nasopharynx. And these are the cells that line that region. They're called epithelial cells. And when it infects these cells, it does go through a normal virus life cycle. It does produce more virus particles. And these viruses that are produced, and this shows virus particles inside cells, these are immature virus particles, and this is a mature one that's about to be released from the cell. But after it's infected this tissue at the top of the throat, it also infects white blood cells, a particular type of white blood cell called a B cell. And these cells travel around the body fighting infection, and they travel through this tissue, and that's how they come in contact with the virus. So it infects these white blood cells, these B cells, and these are the cells in which it enters this dormant phase and in which the cells become immortalized. However, the confusing thing about all of this is it's a cancer-associated virus. It was found in Burkitt's lymphoma. It has this property of immortalizing cells. But actually, when people started screening for the virus in individuals, they found that almost the entire worldwide population are infected with Epstein-Barr virus, and we don't all get ill. We don't develop Epstein-Barr virus, which I will probably abbreviate to EBV, and I probably already have, just to confuse you with acronyms. We don't all get ill. We don't all develop EBV-associated cancers. So why is that? Well, that's because we have a very good immune system. And these immortal EBV-infected cells are seen as foreign by our immune system. And most of them are destroyed. So this is the life cycle of Epstein-Barr virus. The virus enters through saliva. These are these epithelial cells in this region at the top of the throat that become infected, produce more virus particles. They then enter these white blood cells that are traveling through this tissue. They become immortalized, but we have a really good immune response. So other cells called T cells in our blood attack these infected cells and destroy most of them. But some of them survive. And that's good enough for the virus because it wants to be able to survive in these immortalized cells. And then every now and again, the cells become reactivated and it produces more virus. So generally, providing our immune system is fine, and there aren't any other factors that happen to come along to trigger cancer development, we can live together in this balance. And the virus is very good at hiding in these infected cells because it shuts down the production of most of the viral proteins. So it only produces a very small number of proteins that are required to just keep these cells ticking over. And as I mentioned, occasionally it gets triggered to reactivate and produce more virus particles. So perhaps not surprisingly for a virus with these kind of immortalizing properties, As well as being associated with Burkitt's lymphoma, EBV has also been shown to cause a number of other cancers. And these cancers arise from these two cell types. I'm just going to flick back a second. So these epithelial cells and B cells that the virus has the ability to infect. There are two types of cell that it infects, the epithelial cells at the top of the throat and these white blood cells, B cells, that are trafficking through the area. And the cancers that the virus causes derive from these two cell types. So I've already told you about Burkitt's lymphoma, and the numbers at the side here represent the proportion of these cancers that are associated with EBV. So for Burkitt's lymphoma, 90% of these lymphomas are caused by EBV. There is another kind of cancer of that region at the back of the throat, the nasopharynx, and a particular type of this cancer, 100% of these cancers are associated with EBV. The virus also causes other kinds of lymphoma, so Hodgkin's lymphoma and other types of non-Hodgkin's lymphoma, and about 30 to 50% of those cancers are EBV associated. And it also is associated with causing about 10% of stomach cancers, gastric cancers. Okay, so a little bit more about why does cancer develop. So these cells are in our body, our immune system is destroying them. However, they are dangerous to have around. So if there is a problem with our immune system, they have the capacity to grow into an EBV positive cancer. So any severe, and this is severe loss of immune control. This isn't about just feeling a bit under the weather and our immune system might be being a bit down. This is when your immune system is suppressed. For example, if you've had a bone marrow or a solid organ transplant and you've been given drugs that stop your immune system from working because you don't want the transplant to be rejected, or if you're infected with HIV and you're suffering from acquired immune deficiency syndrome, so your immune system is very incapacitated. However, not all EBV-associated cancers happen because of problems with the immune system. Sometimes they develop in what appear to be healthy people. And this is where we come back to the other factors. So I talked to you about Burkitt's lymphoma and us knowing that malaria is a cofactor. But in other cancers, there are often other factors that help to drive the cancer to develop. So this can be genetic changes, environmental factors, or other agents. And for some of these cancers, we have a good idea of what they are. For others, we don't know what they are, and we're still trying to understand that. Okay, so I want to try and take you into a little bit more detail into the kinds of questions that we ask in my research group. And this really centres around how the virus makes these B cells immortal in the first place. So here I've drawn another cartoon. I haven't drawn it actually, I took it from free images online, but anyway. There is a, if I drew it, it wouldn't look like this, I could assure you. Here is a host B cell. These are the chromosomes that are in this host B cell, and this is a zoom in, this is a chromosome, this is all its DNA kind of stretched out. And what I just want to give you is a little bit of a description of what a gene is, because this is going to impact on the next couple of slides. So within our DNA, the regions of DNA that contain all the information that code for proteins. So these are the molecules within our cells that do all the key jobs to allow those cells to function. So in this case, we're probably easy to think about proteins that drive growth or proteins that stop growth. So these genes contain all the information in order to produce those proteins. So what we know is that EBV produces proteins, some of which go to the outside of the cell. I'm not going to talk about those. But most of them go into the nucleus of the cell, and they bind to these chromosomes. They bind to DNA, and they change which genes are switched on and off. And this is really critical to understanding how the virus makes these cells immortal. And actually, by finding out which genes the virus switches on and off to make cells immortal and drive them to develop into cancer cells, it gives us a blueprint of how these kind of B-cell cancers, these lymphomas, could develop in the first place. So it gives us wider information outside of information on just EBV-associated cancers. So this is a scary slide, and if you're not scientifically minded, I don't mind if you just concentrate on this top part and you ignore me for the next minute. So basically, this gives you an idea of some of the projects that we're working on in the lab to try and understand how the virus makes B cells immortal. And the central questions we're asking are, how does the virus make the cells immortal, and how does this drive lymphoma development? So if you want to take that as your take-home message, that's absolutely fine. If you want to try and bear with me and get a little bit of information, that's great too. So we started off, when I set up my group at Sussex, trying to understand how the virus controls the production of these proteins that drive the cell to become immortal. And I've put kind of names of some of the people that have been involved in some of these projects through the years, and you can put some of these names to faces in the latter part of my talk. We then moved on to asking key questions about, okay, so we now have some information about how EBV controls its own protein production. So what do we know about how these proteins then affect the host cells, and how do they immortalize them? And this has been a real central question in our group that has led to us studying lots of different aspects of this. So we've studied how cell cycle is controlled by EBV. We've also studied a set of genes called runks, another silly name, as my daughter tells me. I work on things with very silly names. It's true. We all do as scientists. Every field has a horrible set of acronyms. And both of these projects are really interesting, because by working on EBV and understanding how the virus changes proteins and genes in these pathways, we've discovered that actually these proteins and genes affected by EBV are also affected in lots of other cancers. So it gives us wider information as well. This project and the project where we've looked at different strains of EBV are collaborations with a colleague in London, Paul Farrell. So there isn't just one type of EBV across the world. There are slight variations in the virus in different parts of the world. And we've been studying how that might impact on how the virus immortalizes cells. But one of also linking into this, we've been trying to understand how the virus takes over the host cell. And this is where the structural biology side of work has come in. We're very interested in trying to understand the really fine details of how the virus proteins hijack host proteins. But the two key projects that I'm going to focus on just a little bit more are one of the questions that we then, after kind of asking this main question, set about trying to find out where exactly these proteins were binding in these chromosomes. We know they change genes, we know they change which genes are on and which genes are off, but how do they do it? Where are they going in the host chromosomes to do this? And then this led on to questions of exactly what is the mechanism of that? What are they doing when they get there? So I'm going to try and explain a little bit more about that in a second, but I wanted to come back to this because, as I said, that slide was quite busy, and some of you might be thinking, well, this is all very well, but she started off telling us about all these cancers that are caused by the viruses, so surely what she should be doing is finding cures for these cancers and treating patients. Why is she messing around in the lab asking silly questions about which genes are turned on and off? And that is a perfectly reasonable question. And I just wanted to try and sell the fact that this kind of basic research really matters. So if we're going to be able to find out how disease develops, if we're going to be able to find new ways to stop disease, we really need to find out how the disease develops in the first place, because we can't stop it unless we know how it arises. So if we can find the key steps that drive EBV cancers to develop, we can help find new targets for drug development. That's always one of our aims. It might be a long-term aim, but that's always what we're hoping for. But as I hope I've alluded to a little bit on the last slide, it also tells us more than just what's going on in EBV cancers. It tells us how other cancers can develop and how key proteins involved in those cancers might be working. So that's my little spiel about basic research. And I'm going to try and tell you a little bit more about the kind of basic research we've been doing just in the next couple of slides that centres, as I said, on where do EBV proteins bind to host cell chromosomes and how do they then turn genes on and off. So back to this illustration that I didn't draw of the host cell with our viral proteins inside its nucleus that are binding somewhere here. And this work was carried out by a PhD student in my group, Michael McClellan. And he asked, where are these proteins binding in the host chromosome? And what we found was actually something quite surprising. So you might expect that if you are a protein that wants to turn a gene on, you might associate with the start of that gene, the business end of the gene. And that's true for a lot of factors. But EBV seems to produce factors that turn genes on and off by binding at long distances away. And this is an example of a gene that would produce a protein that drives cell growth. And that's what we know, the virus turns on those kinds of genes. But its proteins are binding a long way away from the start of genes. And actually, this is an example of data from one of the viral proteins we work on. A really small proportion of the binding sites are near the starts of genes. 84% of them, in the case of this protein, are a long distance away from genes. So how's that working? How can they be binding over here and switching on a gene by kind of influencing what's happening here? And that's what we set about trying to investigate. So how can genes be controlled from long distances? By pink elastic bands? Well, of course not. It would be fun if it was pink elastic bands. But this image I quite like because it illustrates exactly what I want to try and explain to you, which is it's by looping. So in that previous diagram, I showed the DNA all stretched out, the binding sites down here, the genes up here. So how are these two parts of our DNA communicating with one another? And they have to come in close physical proximity in order to influence one another. And the answer is by DNA looping. And we know that EBV, we found out that EBV affects the looping arrangements between these long-distance control centers and the starts of genes. So this is our EBV, this is our gene, which is a long way away, not that far away. I haven't got a giant screen, so you'll have to bear with me on the distance. It can be much, much further away than that. The virus produces proteins that bind to these long-distance control centers, and what they then do is they cause looping of the DNA in between the control centers and the gene start, and recruit a lot of other proteins that are already in the cell to enable them, in this case, to turn this gene on. So the virus is influencing the three-dimensional arrangement of the DNA within the cells. And I just want to tell you a little story about two genes that we work on, which have silly names. The first is MYC, and the second is BIM. And MYC, the Vice-Chancellor referred to, I actually spent a little time in my earlier scientific career in Anne's lab working on a different aspect of MYC biology. But I can tell you something slightly different about it. So they've got silly names. What do they do? Well, MYC is a really important gene in driving growth. And we know that the virus switches on this gene. This is the viral protein that turns it on. Don't worry about that. This is the MYC gene, switches it on, pushes cells to grow. Okay? That's kind of a general concept that I've been trying to reiterate. But the cells don't like that very much. And if you produce too much MYC, they do this thing that I was telling you about. They try to commit suicide. And they do this by switching on another gene that's called MYC. So this is this relationship between MYC and BIM. Well, that's no good for the virus. The virus doesn't want this to happen because it won't be able to survive in the infected cells. So what it does is it produces another couple of proteins that switch BIM on. So it keeps driving things in this direction and stops the cells from killing themselves. And as I explained to you before, that's really not a good thing for the host cell, but it's a good thing for the virus. So, and we know actually that in lymphomas, these B-cell cancers that occur without EBV, these two genes are often changed. But the virus has found a way to do this directly. They're changed by different mechanisms in non-viral cancers. So how is the virus doing this? It's switching on MYC, it's switching off BIM, but how? Well, it won't surprise you, given what I just told you, that it's through looping. So what we discovered, and this was work carried out by a research fellow in my group, David Wood, is that actually in cells that don't have EBV in them, this is the MYC gene. The MYC gene is controlled by lots of these long-distance control centers. And when the gene is off, there's a little bit of looping, not very much, to some control centers down here. But when the cells become infected with the virus, all of the looping arrangements around this gene get changed. And the virus produces a protein that binds to these control centers further up from the gene and causes those to loop and interact with the gene and switch it on. Well, this is interesting because we've discovered a new mechanism. MYC is turned on in other cancers by different mechanisms. But what it also tells us is something quite interesting. Aha, it's worked. That I've already explained to you that for Burkitt's lymphoma to develop, you need EBV, and malaria is also an important cofactor. Well, there's actually a third thing that goes wrong in Burkitt's lymphoma cells, and that's a genetic change. And it's a genetic change that happens in this region of the MYC gene. And what we've never been able to understand is when that happens. So does that happen before the cell gets infected with a virus? Or does the virus infect the cell and it somehow promotes that genetic change to happen? And our data would suggest that actually the virus may have something to do with making that genetic change happen, because the genetic change happens in this region where the virus is promoting lots of looping to occur. And we're carrying on with our investigations into this. So this is MYC. This is the growth gene that the virus is switching on. It now wants to switch off the suicide gene, BIM. And it does exactly the opposite in this case. So if looping is a good way of switching on genes, disrupting looping is a good way of switching off genes. And that's exactly what happens at BIM. BIM is making lots of looping contacts to all these long-distance control centers when the virus isn't there. When the virus is there, it disrupts these loops, and all of these control centers get coated with a silencing protein that keeps the gene off. Well, what's really interesting about this is this silencing protein is around in the cells and is doing lots of silencing of other genes and goes wrong in lots of other cancers. So there have been drugs developed to block it. And if we treat cells, EBV-infected cells, with these drugs, we can reverse this. We can push the loops to occur and we can switch this suicide gene back on. And this is a measure of cell suicide. So when we treat with this drug, we can make the cells commit suicide. So this is a really interesting kind of early example of how we might be able to think about using drugs as a result of some of the basic mechanism discoveries that we've made.